So we'll start. Good afternoon, everyone. So today we will discuss the second class on science and tech. Yesterday what we did? We did most of the questions from general science as well as space, communication, defense related questions. So today our major focus will be on healthcare, biotechnology and a little bit of emerging technologies. So how are you preparing science and tech, or what is your approach to science and tech? PYQs, okay, then some compilations. If you see recent question, if you especially look at the 30 question which we are going to discuss today, most of these questions are from very themes which you are very familiar with. You can see so many questions related to COVID-19, so many questions last 3 years related to vaccine development, so many questions related to immune biology or technology behind that kind of sciences. So many of these questions are very familiar themes. If we look back from now, there is a mistake that you can make in analyzing the PYQs. You will see this question, this is from COVID, this is from related to DNA, gene editing or something like that. These are very easy questions. Maybe looking back, we will find that these are very easy questions, or everything is asked from something very much we heard of. But during the exam or exam time, relating this with these questions are as such not that easy. What is difficulty of these questions is, even though most of these are from very familiar themes, what they asked is not that familiar topics. They are asked from familiar themes में fringe topics है, which is we usually neglect. From that areas questions are picked. Even though so many questions from COVID-19, how many of those questions you can answer with confidence is very less. COVID-19 you will see in every compilation, every current affairs XYZ you are learning for this exam, you will find that particular topic there. But finding what exactly asked in the exam, that becomes really a challenge. Now how to address this? What you can do for, what you can approach you can keep for science and tech in general to address these kind of questions, or this is the trend that we are seeing in the recent past. We are anticipating similar trend will be there in the future also. Question will be from topic themes which are very familiar, but the nature of the question will be little bit tilted or little bit difficult. How to address this? Tell me.
This year I am making a compilation of 1 year Hindu science and tech, one writer.
Decent. You are making a compilation yourself. Good. One thing is, yesterday also I told you, there are two different types of topics. Whenever you are reading any XYZ materials, there are some topics like where you only need the peripheral knowledge. For example, we did a question on ELISA yesterday. What all you need to know is, what is this? What this XYZ program is for. Your knowledge should end there. Digging more on this is not relevant for our exam. At the other time, when you are studying something related to mRNA vaccine, if you are learning something related to mRNA vaccine, recently COVID vaccines have been formed. This is a totally new technology that has came into existence. So when you are learning this, you have to go much beyond this. You have to go much beyond the peripheral knowledge. You have to really work on it. Read from multiple sources. If you depend for these kind of topics, where it went? Yeah, for these kind of topics, if you depend only on one XYZ magazine, then you are always going to be lagging. For these topics which are so much in news or some major developments and happen, and you are anticipating that this is a very important topic which can come for the exam. At least for these topics, you have to prepare your own notes. Depending on any magazine, that becomes very risky by looking at the nature of the question that is being asked. That is one approach. Whatever topics you are learning, try to divide it into 2 topics. At least identify 30-40 themes from science and tech, which you will anticipate a question in the upcoming exam. Not just PYQs, from current affairs material, from PYQs also, something which is very relevant that is going on in news. That kind of topics you list down, at least 30 topics from science and tech, and prepare that very well so that any fringe question also you can at least try to attempt it. This is one approach. What else? Another thing which you see in today's discussion is so many questions are on application level. Yesterday also we have seen so many questions on application level. Now how to address these questions? Many times if you exactly know the application, you know the process, you can answer it. Otherwise what we will do? You have to have very strong base. If you exactly know how a cell functions, where is the DNA placed, what is the function of DNA, what is the function of RNA, maybe you don't get a question directly from what is DNA or RNA, that much basic question don't come. But even in advanced questions, for eliminating a statement, for identifying what is wrong in this statement, what is by idea or by logic this statement cannot be true. To make that conclusion, your fundamentals should be very strong. At least in this biology topic, at least in this healthcare, biotechnology related topics, the fundamentals of your conceptual clarity should be very best. So what to address, how to do this? You can, don't neglect this. What happened in science and tech preparation, we'll take a current affairs compilation and we'll limit our study there. And we don't have any very good book, standard book for science and tech. At least go through NCRTs. At least go to the 9, 10 and some chapters of 11 and 12 NCRTs. If you are weak in science, if you are from arts background, then this is must for you. If you are very strong in science, you are having a background in science, engineering and all this, you can still be a little bit light on it. Otherwise, you should do this. Maybe you will not get a question directly from this, but so many application level of question today also you will see. How we answer this is we should have very strong fundamentals. So this is some kind of approach you should keep for science and tech. Go ahead. So we'll start with today's discussion. So we have 30 questions, same. What we did yesterday, same format we will continue. Take 30 seconds and answer this first question. Okay. What is answer? D. They protect body from diseases caused by pathogens. Any other answer? Both of them. D answer. Okay. So which one of the following statement best describes the role of B cells and T cells in the human body? What is B cells and T cells? What basically fundamentally are they? Both of these are, leave it. Both of these are types of WBC. And what is WBC? White blood cells. What is the function of WBC? Blood clot basic function is to provide immunity to the body. How they provide immunity? There are different and kind of mechanisms. What these B cells and T cells do is they produce antibodies. Specific proteins that related to antibodies, B cells will directly produce these antibodies, and these antibodies will go and fight against whatever the external component is there in the body. Now what T cells do? T cells will regulate how much antibody should be produced. What are the other options? Why this question came? Herd immunity related topics were in news for some reason or the other. Even related to COVID, immunity is so much of discussed topic. Like some population is not being affected, whereas population in other countries are severely being affected. So somewhere or other this was in news. Even in vaccine technology you will see what our purpose is to produce enough number of antibodies by different ways different vaccines work. Objective is to produce different kind of antibodies. Now we will study about antibodies, we will study about immunity, but exactly T cells and B cells means it's a very specific topic. Go ahead. Now white blood cells is not just B cells and T cells. There are five different kinds of basically white blood cells. Each of these will resist this foreign body or an antibody that is entering in the body in different ways. Just at least have familiarity with the word. The first one is neutrophil. What it does? If a new body is come, it will engulf it and try to digest it. By digestion, it tries to destroy this foreign body. Second part is eosinophil. That is basically related to allergic relations, if inflammations in the body, related responses. Now, basophil is the very similar kind of thing. Then we have lymphocyte. The B cells and T cells are part of lymphocytes. These are the components which will directly produce the required antigens. Now monocyte is like specialized kind of blood cells for XYZ applications. No need to learn too much of it, but at least what is the function of WBC, function of RBC you should be clear of. What is function of RBC? To distribute oxygen, to carry oxygen. That's why it is red in color. Why it is red in color? Basically iron is there, other pigments are there, and the main function of RBC is to carry oxygen from lungs to all other parts of the body, while WBC function is to provide immunity to the body. What happens if WBC is produced in uncontrolled manner? Which kind of disease you associate with the process of WBC being produced in uncontrolled manner? Now it started attacking the good cells of the human body also. What disease you associate with this? It is cancer. Most cancers is one way or the other uncontrolled production of these WBCs. Now this will attack specific part of the body and try to destroy that organ. Go ahead, answer the second question first. हो गया? बताओ क्या आंसर है? It's very easy question. What is answer? D. B. 3 only, okay. Consider the following statement. It is also a 2022 question, last question also. 2022. Other than those made by humans, nanoparticles do not exist in nature. Okay. Nanoparticles of some metallic oxides are used in the manufacture of some cosmetics. Nanoparticles of some commercial products which enter the environment are unsafe for humans. What is general thinking on these statements? Suppose I am not well aware of nanoparticles. How should I should go with this question? That only happens if I know it. Suppose I am not sure about what nanoparticles is. How should I approach this question? See this option. Nanoparticles of some metallic oxides are used in manufacturing of some cosmetics. Cosmetics is broad area. Now some cosmetics, some metallic oxide means it is also asking application of a technology. And there is very broad or generalistic statement is made. So my high tendency is this should be right. Now come to third option. Nanoparticles of some commercial products which enter the environment are unsafe for humans. What is nanoparticles? There either some dimensions will be in the nanometer range. It is not required the whole particle should be in nanometer range. Some dimension which give them XYZ characteristics will be in nanometer range. And nanometer range means at least 1 to 100 nanometer. What is 1 nanometer? 10 raised to minus 9 meter. So if any of one dimension is coming under this, we call it as a nanoparticle. And why this is important? Why basically the properties of material changes at nano level? You see that gold is yellow in normal crystal gold. The same gold, if you make it as a nanoparticle, it is pure white. Why it is changing its properties? Why? Guess karo, at least if you don't know. What happens if you make it is into this much small particles? What happens? Surface area will be extremely high. Normal, the substance cannot have anticipate this kind of surface area. This will become at some molecules level. So at this level, the surface area will becomes very high, and this property will give XYZ requisite properties to the particles. And the property of these particles at nano level may be completely different from the crystalline metal. We told about gold, yellow, it changing into white. Maybe these electrical properties, magnetic properties, all these properties are being impacted by changing this size of the particles to the nano level. That's why this technology is finding application in many areas. Now come to the first part of the question. Other than those made by humans, nanoparticles do not exist in nature. Can this be true or false? There is, if you have read newspaper, there is a view or there is some articles referring viruses as nanoparticles. What is viruses? What is virus actually? Are they living or non-living? Viruses are considered as non-living organisms. All others, bacteria, fungus, all these are living organisms, but viruses are considered as non-living organisms, and they don't have an existence of its own. They can only exist inside body of some XYZ, inside body of human cell or inside body of XYZ other cells. By utilizing the nutrients from the cell, it will live. It will replicate into many parts, many more viruses. But of its own, it cannot live or it cannot sustain in the environment. So there are references of viruses as nanoparticles. Now nanoparticles are also major pollutant. Where we see this pollutant? In volcanics, volcano eruptions. Even nano level dust particles are also emitted in volcanic eruptions. If some of this you can connect with nature, there is a possibility, there is more possibility that some particles in nature can take off nanoparticles form. What is? Which one? Viruses. Yes, yes. It's not something which is created for this question. There is viruses are also categorized as nanoparticles. So if you remember any of these aspects, or even if you look at the statement, there is more possibility that at least some XYZ in the nature which can take the form of nanoparticles. Well, these two statements are very generalistic statements. If I know I answer this question correctly, if I don't know the answer, my approach should be to go with these two as correct. And nanoparticles is some kind of powders. Assume it as a powders. Then these powders will obviously create some environmental problems. Even microplastic we have learned in detail. What are microplastics?
Plastics are naturally or artificially broken to very small.
Very small, 5 mm, less than 5 mm size particles, plastics broken down into less than 5 mm size is nanoparticles. These particles are creating so much of environmental problems, ocean pollution, whole ecosystem is being impacted by this. Now assume what will be the level of pollution if nanoparticles have been used in a mass level. So environmental pollution should be there. These two options I don't find any reason to eliminate, even if I don't know nothing about nanoparticles. Go ahead. So here answer is 2 and 3, answer is D. Answer the third question first. If you are watching online, you can answer the questions. You can mark ABCD in the live chat. Okay. Tell me what is answer? D. DNA barcoding. How do you answer this? What is DNA barcoding?
May be related to that you are categorizing your barcoding.
Any other options? DNA barcoding. It is basically you are taking a small part of DNA, then you are considering like studying it and studying from which species this came from, all XYZ characteristics of this species. You are matching it with which data you have, and you are analyzing the species diversity of species XYZ about it. So that is DNA barcoding. Now what is the options available here? Assess the age of a plant or animal. Distinguish among species that look alike. Identify undesirable animal or plant materials in the processed foods. So how you go with these options? Think like something which DNA barcoding means something it is, something it should do with genetics, something it has to associate with genes. Now where genes play an important role. Where genes play an important role. This is assess the age of a plant, you told it is carbon dating. We have other methodologies, taking at the rings of the plant. Genes of the plant or animal is not that much of a factor in assessing the age of a plant or animal. Now this, it will be same genetic material will be remaining like that. So it's not, you cannot associate this characteristics directly with gene technology. Now come to this, distinguish among species that look alike. It's very much possible. It's gene, it is using genes you do this. All forensics. What is forensics? Not necessary, it is in criminals only. Studying genetic material and identifying which person is this, which person is. All this is done using genetic data itself, DNA code, matching DNA, something like that. So DNA barcoding can have a very much familiarity, or this can be a very much good application of something associated with genetic. Now identify undesirable animal or plant materials in the processed foods. If you read the news, this is exactly application of DNA barcoding. If you know the news, like if you have collect so many animal species from the seafood and marine exploration, to categorize them, the DNA barcoding is first time used or widely used so far. So this is like a direct application. Even if you don't know this, identifying undesirable plants or animals in processed food means that is very much related to distinguishing species of that look alike. They are like synonymous options. So best possible solution is go with 2 and 3, that is option D. Go ahead. Answer the fourth question first. What is answer? This is little bit very factual question. There is nothing much you can do about it. What is answer? A. Any other answer? D. Correct answer is D. All of these are open source digital platforms. What does it mean by open source digital platforms? Yeah, open source digital platform means, if suppose we made this platform CoWIN platform. What CoWIN platform is for? Vaccine distribution or vaccine mechanism. Now in CoWIN, how it is made, the core code data that will be openly provided plus the license of it. And what is the implications of this? Any XYZ person can use this code and make other applications based on this platform. Open source code. That is basic idea of open source code. Now this exactly, you will remember some of these CoWIN applications were came when the vaccine is having available, it will inform you. There were some websites, some application developed for this. Even though how it works is very difficult, it's very hard to really function it. But there were applications developed for this. These applications are not developed by government or agencies or government authorities. These were done by private agencies. So if you remember something like this associated with all other things, you can relate it and find what is the correct answer. Otherwise, this is a very factual question. And what is this Diksha platform? It is for online education. Who launched it? Mainly it was for during the COVID times, it was more relevant. Who launched this? NCRT, that is under Ministry of Education. Go ahead. Answer here is 1, 2, 3 and 4. Answer the fifth question first. Okay. What is the answer? Batao. See, you're following them or finding your own? What is probiotics? You might have seen this. So many food products you will get something like this: probiotics dahi, probiotics XYZ. What exactly is this? I think we have reached somewhere else. Probiotics basically means good microorganisms. Very good, very good. Probiotics, pro is something positive, biotic means something life. So something like similar meaning it is coming out. Good microorganism, good bacteria that is inside your body, that collectively is called probiotics. Now what is probiotics? Food means food that contains these XYZ bacteria. These bacteria are already present in your body. Now with the XYZ food, it will be stimulated, more bacteria will be there. And what is the benefits of this? What is the functions of the good bacteria that is inside the body? To help digestion, to regulate some production of some hormones or enzymes. These are different functions, basic functions of probiotics or good bacteria that is inside your body. This much clarity you have. This question is very simple now. What are the options? Probiotics are made of both bacteria and yeast. Both these options are correct. Yeast is the dahi which I told you. It may be used there. Bacteria, that is also, bacteria are, so many good bacteria are part of this, our body. The organisms in probiotics are found in food we ingest, but they do not occur naturally in our gut. This is something an extreme statement. If something is totally not in your gut and you are intaking it, there will be some other responses. Like we discussed about antibodies, we discussed about allergic responses by this body. These are not that kind of bacteria. These are within you and we are intaking it just for supplementing it. So this option is wrong. Probiotics helps in digestion of milk sugars. One of the basic major function of probiotics is improve the digestion process. Now milk sugar is something which is hard to digest. Especially people will have this lactose intolerance. So for those people, this probiotics is a very good substance to take to some kind of maintain this lactose intolerance. So this is direct application level. This is also something which are the basic definition of probiotics. All these statement, if you look at this, are developed one way or the other from the basic core concepts of what is probiotics. What is answer here? 1 and 3 itself. Go ahead. Answer the 6th question. This may be one of the easiest question you will get in last year's paper. Okay. What is answer?
B.
You are saying? A. You are also saying A. Saying B. In the context of vaccines manufactured to prevent COVID-19 pandemic, consider the following statements. Serum Institute of India produced COVID-19 vaccine named Covishield using mRNA platform. Who is saying this is correct? We have different types of vaccines. We just now discussed that mRNA vaccine is totally new technology. This is the first time we are using this technology for development of vaccines. This is a totally new platform that is came into being. Maybe you are forgot this because it's 2 years down the pandemic. But at least that time you are giving the exam, you should be very aware that is only 2 major vaccines are there using this technology. Which are those vaccines? No. That is both not Pfizer and Moderna. These vaccines are only at a very limited level we have access to this. Mostly in US and all, these vaccines are in use. And these vaccines both are based on the technology of mRNA vaccine technology. Our Covishield, Covaxin, all these vaccines which we find in India Sputnik vaccine, all these vaccines are not based on mRNA technology. So this option is absolutely wrong. Now this is just game of elimination. Only one option is left. 2 and 3 is correct. Otherwise it's very difficult. Sputnik V vaccine is based on vector based platform. This is very much correct. But remembering every vaccine's technology is little difficult. But at least this much you should have remembered that mRNA technology is related to this because this is a new technology. Covaxin is inactivated pathogen based vaccine. Both these statements are correct. What we should learn more is what are different types of vaccine. Tell me some different types of vaccine. First one is inactivated or killed vaccine. What does it do?
Part of that disease virus is taken, it is made inactivated.
What does it mean by inactivated or killed? Suppose we are taking a XYZ virus, we remove certain parts of it so that it cannot replicate. Now, if it cannot replicate and we are introducing it into our body, what will happen? Same process that we discussed in the first question. We get a new foreign particles come into our body, antibodies will come, they will produce antibodies, then they will white blood cells, all this function will happen and our body learns to destroy this. Now once this virus is destroyed, our body learns the mechanism how to destroy this virus. Now when actual virus come, they can easily identify and produce these antibodies very fast. Now, so that you can prevent infection. What are some examples of this kind of vaccines? Best example is polio vaccine. Other example, hepatitis A vaccine, rabies vaccine are all based on this inactivator or killed vaccine technology. What is other second type of vaccine? Live attenuated vaccine. How this vaccine works?
The organism is spoiled live, it is not killed.
Here what we'll do, the ability for this virus to replicate, we are completely cut off. Here we are not completely cutting off. It is something like you are infecting the human body at a mild level. You are providing a mild infection to the body. Providing a mild infection means live virus is introduced to your body, but at a very low dose such that it doesn't create health issues. And when this infection is there, same process, WBC will be there, antibodies will be generated. Body over the course of time learn to respond and fight and kill these viruses. Now what is risk associated with this? Sometimes the virus will reproduce at a larger level and infect the body. So there is always a risk of reinfection from these vaccines. What are famous vaccines for this? Measles vaccine, rubella vaccine are based on this vaccine technology. What is another type of vaccine? Subunit, you will find it so many names. Subunit vaccine, recombinant vaccine, or conjugate vaccine. How this vaccine functions? Tell me. Don't say DNA code. That is totally different type of vaccines or different types of approach. Here we are introducing fully inactivated or non-reproduction type of virus. Here we are introducing some live virus. Here we are introducing only some parts of the virus. Full virus is not introduced, only a certain parts that is introduced. Now which part we will introduce? If COVID vaccine is there, we will introduce these spike parts. Now if spike part is introduced, what will happen? Same process, body will learn to how to handle these spikes. Now when original virus will come with these spikes, body might have already known how to handle the spikes and they will attack this virus fast. So what are some examples of these kind of vaccines? Human HPV vaccine, human papilloma vaccine. This is a major example of this vaccine. Idea is only a part of the virus is only introduced to your body. Now what is another type? mRNA is there. What is mRNA? Messenger RNA. mRNA is messenger RNA. What it does? Tell me. Tell me more. What is function of DNA? I'm asking DNA. DNA is a double spiral structure. You cannot draw. You will say double stranded structure we will get. And what is the major function of DNA? That is to pass the genetic information. Your XYZ genetic information will be stored in DNA, and this information will be passed to the next generation. Now what RNA does? DNA we have double strands. RNA we have single strand. And DNA will be like breakdown into RNA. And what does RNA contain? Single strand means it is the order to produce XYZ amino acids or protein. So DNA has your genetic information. Now this genetic information may be like produce this color of skin. That is a genetic information. All skin color are coming from genetically related. Now produce this kind of skin is genetic information that is stored in DNA. Now skin will be produced at certain part of this body. Now giving instruction to that skin producing organ, produce these characteristic type of skin. That message is being transmitted through RNA. Taking this order is the major function of the RNA. Taking this order from genetic code to whichever body part that is producing XYZ organ or XYZ doing that activity. DNA is double stranded. Its function is to store the genetic information and pass on it to the next generation. Now what mRNA vaccine does? Tell me. In COVID-19 we seen that this is the structure of this virus. We introduce a mRNA, that is this instruction into the body, and this instruction will be to produce these spikes. These spikes are not introduced. we are introduced an RNA that give instruction to human cell to produce these spikes. Now when you take the vaccine, what will happen? Body will give the order to produce these spikes, and these spikes will be generated in your body. Then the whole same process will happen. Antibodies, new particles came, antibodies will be produced. Now once these spikes are killed, body will learn the process how to handle these spikes. When original virus come, it can very quickly replicate it. So this virus is nothing related to, like, totally different from what we have done so far. It is only introducing the order to produce these kind of XYZ. They are like developed from COVID virus genetic material only. What is the advantages of this? No reinfection like this. These risks are completely avoided, because even this virus is not even seen in the picture. Only one genetic, only small part of genetic is only introduced into your body. So no risk of reinfection. What more? That is not the major advantage. What is the major advantage of mRNA vaccine? Why we are celebrating a new technology has came? Because this is like a platform. This is like a platform. Now, this vaccine development takes decades. You have to develop the vaccine, you have to test it on n number of individuals. This whole process takes decades. Polio vaccine is developed after study for taking many years. mRNA vaccine means you have access to a technology where you can develop, make changes very fast. So vaccine development process, the whole process is like compressed into a matter of one research lab. What earlier was happening over decades, now we happen within months in a research lab. So vaccine development, this is now only available for COVID. If we expand this, maybe more number of diseases can come under this, more number of vaccines can be brought into this. And what is one more advantage is, since this process is very fast, if COVID virus, we have seen that the virus itself is changing its genetic component. Like new Omicron, different variations are coming. So these vaccines are not able to address these changes fastly. While mRNA vaccine, we have a tool to even address the sudden changes that is happening in this XYZ virus. So due to these factors, mRNA is a celebrated technology in vaccine industry or vaccine development. And what is another type of vaccine? That is DNA vaccine. Like in RNA vaccine, we are only introducing this instruction. In DNA vaccine, we will introduce this complete double-stranded DNA structure. Now, which vaccine is famous for this? One example. There are no vaccine that is so far developed with DNA vaccine technology. That is like still in research development. Some vaccine might come in near future. Technology is getting matured, but so far we do not have a commercially used vaccine with DNA technology. So these are basic how vaccine works. Now question can come from mRNA as well as DNA vaccine technology. These are the new guys. So go and learn, like these two topics you should keep in which were themes I was discussing, at least 20-30 themes from science and tech. mRNA vaccine, even though it is little over, we are out of COVID times, still mRNA vaccine is in use for one reason or the other, as well as a DNA vaccine. Do learn this very smartly so that even a fringe topic something is asked, you should be able to answer it or address this at least.
In case of DNA virus vaccine, the DNA virus of that particular virus will be introduced into the body.
It is not like full DNA is taken and introduced to your body. There will be some editing done on it so that your DNA will not be affected, or it will not become another virus source in the body. So some editing will be done, and only then after this DNA virus or DNA vaccine will be developed. All advantages we associated with mRNA vaccine, similar advantages is also there with DNA vaccines also. Think
it gives command.
we have this some picture. Messenger.
mRNA it gives command to body.
mRNA will give command to the body to produce this kind of protein, spike protein. Now this spike protein will be handled by XYZ antibodies. Then body will acquire that XYZ characteristics. So this is diagrammatic representation of what we have discussed so far. Understood the concept. Now basic details you need to, this is not enough, this is just peripheral picture. Questions are coming from fringe topics. So you need to go back and learn this from Google search, you do some good material and learn something more about it in detail and make a small half-page note of it. And don't, at least for 30 themes which you very important theme you identify from science and tech, you think this can come for the exam, don't depend on one institute material. Make your own notes, make your own some effort and make it. Otherwise it's not going to be sufficient considering the nature of question that is being asked. So that's it. Answer the 7th question first. Wait. हो गया? What is answer? B. Who are all saying B? Who is saying C? 1, 3 and 4. How you approach this question? Which of the following are reasons for occurrence of multi-drug resistance in microbial pathogens in India? Genetic predisposition of some people, taking incorrect doses of antibiotics to cure diseases, using antibiotics in livestock and farming, multiple chronic diseases in some people. Select the correct code. How to approach this question? What is this multi-drug resistance?
HDRT.
What does it mean?
We have several lines of antibiotics.
We have several lines of antibiotics. If our first line of antibiotics, the organism acquired the capabilities to defend that antibiotics. That kind of XYZ characteristic we called as multi-drug resistance. Now, who is acquiring the characteristics? The XYZ microorganism. Whether it is for tuberculosis, it will be tuberculosis bacteria. Mostly it is associated with tuberculosis. So who is acquiring the characteristics? Whose characteristics are being changed? That is, this microorganism characteristics are being changed. Now here look for options where you can associate this property with this. This has limited things to do with who is taking the medicine. It is more with what the microorganism, how it is undertaking the changed atmosphere or changed environmental conditions. Now look at the genetic disposition in some people, how it will result in multi-drug resistance. At worst, what it can do, maybe the drug will not be applicable in XYZ person. So he may be treated with some other drugs, or there may be reaction for if certain drugs are being taken by a person. These all can happen due to genetic predisposition, but it is unlikely that the characteristics of the germ will be changed. What is second? Taking incorrect doses of antibiotics to cure some diseases. Incorrect doses means what will it does? It will directly change the environment of this microorganism. First it will get this virus. Antibiotics, if usually it happens If you go for, even for a fever, if you are written this antibiotics, it will be for 4-day course, 3-day course. And this whole virus in your body will be killed only in this period of time. Now, if you take 1 day and you left the vaccine, what happened? You may get relief for that day, but this will not be totally removed from your body. It will remain in your body, and over a period of time, it will acquire the characteristics to defend this XYZ antibiotics. Using livestock in antibiotics in livestock farming. This is same as what we discussed here. It's not using it, its problem is misusing it. You are giving this XYZ antibiotics for this growth hormones, all these are injected into this livestock for getting more milk, for getting more meat and all this. Misusing this is creating the problem. If you are scientifically using it, it will not affect it. But people are not aware of it. Maybe higher quantities they will inject, or they will not follow the XYZ instructions how to use these antibiotics and all. So this is same atmosphere that is created in human body can be created here also. Multiple chronic diseases in some people. This is also related with genetic disposition. Sometimes if it chronic diseases, effectiveness of this drug will be reduced. But very small chance is there, it will also change the characteristics of this drug and make a new type of organism. That chances are very less. So most feasible, most right option you can select here is 2 and 3 is right, that itself. Go ahead, answer the 8th question. What is answer? All of you are answering B. D. Any other option? How to approach this question? It is very easy if you know everything about hepatitis B. Suppose we are not that much aware of hepatitis B or hepatitis C, whatever it is. What is hepatitis? Which organ it impacts? Liver inflammation. Is basically hepatitis. Now we are trying to find those options which are likely or unlikely to go with. This is one of these questions I find extremely difficult if you don't know. What is first option we have? Hepatitis B is transmitted much like HIV. That is it sexually transmitted or not? Or from genetically mother to, not genetically, by breastfeeding and or mother to baby, is it transmittable or not? That is what is asked. Now, hepatitis B, unlike hepatitis C, does not have a vaccine. Let's put this on hold. Globally, the number of people infected with hepatitis B and C virus is several times more than that those infected with HIV. What you can infer from this? At least I can say that several times is given. It is not given 2 times or 3 times. 2 times or 3 times is given, I have more tendency to take it as wrong. It is given that several times. So maybe that's also like a very exaggerated guess. You cannot take it as. Some of those infected with BC viruses does not show symptoms for many years. These are very technical options. It's only answerable if you know this. What is correct answer here? Hepatitis we discussed in last question. Hepatitis is having A, B, C, D, E, different kind of viruses. In last virus, when we are discussing vaccines, live vaccines are applicable for hepatitis A. Similarly, hepatitis B also has vaccine. Only hepatitis A and B has vaccine. C, D, E don't have vaccines. Yes, not correct. Unlike hepatitis B, unlike hepatitis C, does not have vaccine. B does have vaccine. A and B does have vaccine. C, D, E and 3 types doesn't have vaccines. So this is the best feasible option you can take. Don't too much supply mind or too much go for guessing. Cannot answer, leave it. That is also a strategic part of your answering the paper. Before, yes. So many, like even TB test you will do, so much HIV test also you will do. Something if you are aware of, then go with eliminating 1-2 options. Then if at least you can 2 options you can eliminate, then I will support you go for 50-50 guessing. That is reasonable. If you don't know anything, then sitting and guessing means it is very risky. It's more risky than leaving the question. Answer the 9th question. This should be an easy question. What is the answer? These are those questions, so many questions you get that application of XYZ technology is what? In this question, only eliminate those which you are 100% sure that ye ho nahi sakta. Otherwise, even an exaggerated connection is there with this XYZ, what is asked and the technology. Take it as there will be some connection. These are like that kind of a question. What is it asked? Plant disease. Plant disease is impacted by all of this. There will be wind. If you have done some kind of aquaculture, some kind of farming in home, plant infection is easily spread through all diseases. One plant it can go from rain, it can go with bird, wind, all this happening. So here answer is only eliminate an option if you are very clear about it. Otherwise, take it all as by default right. Here answer will be D. Answer the 10th question. It's okay if you are not able to remember this, because this was during that time, this was a major issue. 2017. So we'll discuss some general things. What is the answer? Roughly. Who is saying D? B. You are saying C. Answer is C. So it is about, consider the following statement. In tropical regions, Zika virus disease is transmitted by same mosquito that transmit dengue. Which mosquito they are talking about? Aedes aegypti. Which are diseases it spreads? Zika and dengue is there. Very good. What you told? Chikungunya. Chikungunya is there. Yellow fever is there. Dengue is the famous one. All these diseases are spread by same XYZ vector. What else? Second statement, sexual transmission of Zika virus is possible. This is also possible. What is impact of Zika virus? Problem is you will get very mild symptoms like simple fever, rashes in the body. Symptoms are not that big, you will get cautious and take the treatment. So symptoms are very mild, but the consequences can be very harsh. Like even genetically defected babies are born for people infected with Zika virus. So that is the implication. And what is the treatment for this? There is no clear treatment methodology for Zika virus. Go ahead. It is happening in, yes, tropical region, basically Africa and all, South America also. Answer the 11th question.